everyone, I'm Daisy Liu and I'm a second year student in Dr. Diehl-Jones' lab. And the past 12 weeks have been spent looking at this question: is BNP3 regulated by hypoxia and oxidative stress in human intestinal cells? So I'm going to start off with some background information, including some of the research that's been done in our lab so far in the previous years, and then move on to the experimental methods and results that help me answer my question, and finally some future directions that I intend to pursue. Okay, so previously in our lab, it's been shown that preterm infants given human milk fortifiers actually experience higher oxidative stress and increased BNP3 protein expression. And BNP3 is a cell death gene, so that's obviously not a good thing. It's also been shown that misoprostol, a drug, abrogates this effect. So what exactly is the clinical link for these findings? BNP3 is actually associated with necrotizing enterocolitis, which is a really serious inflammatory condition of the colon, and that's experienced in about 10% of premature babies. So BNP3 is part of the BCL2 gene family, and it encodes a mitochondrial protein that acts to induce apoptosis in many cell types. What's interesting is that it actually contains a hypoxia response element in the promoter that targets the HIF1 alpha pathway. So HIF is hypoxia-inducible factor, and that's a transcriptional factor. And you can see the gene located here on chromosome 10, and this is the molecular model. So premature babies experience a myriad of complications, including profound hypoxia, and it's been shown that hypoxia and oxidative stress are linked to many of these diseases and conditions that they experience. Furthermore, hypoxia and oxidative stress have been shown to induce BNP3 expression. However, it's not been shown thoroughly that BNP3 expression is investigated in HT29 cells. And that happens to be the cell line that we're looking at. So this is to reiterate a schematic that shows what I just said. Hypoxia or ROS act through the HIF1 alpha pathway to affect BNP3, and ROS can act through other pathways to affect BNP3 expression that ultimately leads to cell death either through ER necrosis or mitochondrial apoptosis. So the drug that we looked at is called misoprostol, and we looked at it because it's been shown that it abrogates some of the effects of BNP3 expression. It's a prostaglandin type E analog that's been used for treating gastric ulcers and other applications. So just to reiterate again, it does have effects on BNP3, but we're looking at it in HT29 cells specifically. So back to the cell line. We find it special because it provides us with a homogeneous population to experiment on, and not only that, but it constitutively produces mucins, which makes it a very physiologic model to look at. And also it's an adenocarcinoma cell line, human cell line. So these are my three hypotheses. I'm going to show them in a schematic form so it's easier to see. Firstly, I hypothesize that oxidative stress upregulates BNP3. Second, I hypothesize that hypoxia also upregulates BNP3, and that misoprostol modulates BNP3. And these are all specifically for HT29 cells, if I haven't made that clear. Okay, so there are several endpoints to my study. I decided to look at oxidative stress through a ROS assay because that's indicative of the amount of oxidative stress each treatment causes. And I also looked at BNP3 protein and mRNA expression through Western blotting and qPCR. In an attempt to look at what kinds of mechanisms might be affecting BNP3, might be an effect of BNP3, I looked at TMRM microscopy, which investigates mitochondrial membrane potential. So the first of my Western blots was completed to look at what time frame leads to the induction of BNP3. So I treated with hydrogen peroxide and waited up to 4 hours, and you can see that the effects are time dependent, and the maximum induction occurs at about 4 hours. So we decided to stick with that and looked at a biological triplicate of these time points. You can see that there are 3 sets of proteins, 3 sets of cell samples. So this is set 1, set 2, and set 3. And you can see clearly that there's greater protein expression after 4 hours compared to the 0-hour control. And the next Western blot I did looked at the effects of hypoxia and misoprostol. So it's clear that treating with hypoxia increases the amount of BNF3 expression, but it's interesting to see that misoprostol actually decreases the amount of BNF3 expression, which is as expected. So I decided to confirm my Western blot results with qPCR or real-time PCR, and you can see that they are consistent. So hypoxia again has the highest level of BNF3, lowered by misoprostol once it's added, and these are my hydrogen peroxide time course treatments. So you can see at the 4 hours it shows higher BNF3. Okay, so the results of my ROS can be a little daunting to look at. I've outlined the different conclusions that we came up with after looking up at this figure. So first and foremost, just by adding hydrogen peroxide, we can see that it causes a significant increase in the amount of ROS generated, or oxidative stress, if you will. There are 4 lines overlapped down here, and the only difference between them is whether they're treated with normoxia or hypoxia or with misoprostol added or not. So clearly that the addition of misoprostol does not affect the amount of oxidative stress generated, which is also as expected. Now what was surprising and really interesting is you can see that there's a significant difference between cells that were treated with hypoxia and peroxide and cells that were treated with normoxia and peroxide. So hypoxia doesn't actually increase ROS. In fact, it generates much lower ROS than we expected. So this tells us that because it generates lower oxidative stress, the increase in BNF3 expression is likely not caused by oxidative stress, but rather hypoxia, and that leads us to believe and look at the HIF1 alpha pathway more. And just to recap, it seems that in our HT29 model at least, the hypoxia is the main regulator through the HIF1 pathway that affects BNF3 expression. ROS through other mechanisms has been shown to affect BNF3, but not so much in our model. And everything else here just illustrates how complex the story really is. There are different isoforms of BNF3 that are regulated by phosphorylation that cause different effects in other cell types. And it's not very clear right now, so I can't give you the full story, but it's been really interesting to look at so far. So one of the other endpoints I looked at is TMR and microscopy. Because BNF3 affects mitochondria, we decided that maybe mitochondrial potential is also affected. So basically that involves transfecting cells with different BNF3 gene constructs and looking at the difference in mitochondrial potential through microscopy. And it's still a work in progress, but it's really interesting to see what that turns out like. In conclusion, I've found several things. Hypoxia and oxidative stress induce BNF3 expression. And not only that, but adding misoprostol to hypoxia-treated cells decreases BNF3 expression. And finally, according to our ROS results, hypoxia appears to increase BNF3 through the HIF1 alpha pathway because oxidative stress doesn't. So in the future, I'd like to confirm our findings because our data is preliminary at this point. I'd also like to investigate further mechanisms of BNF3 and evaluate the effects of phosphorylation on the protein. So a huge thank you to our research associate, Dr. Yan Hai, for her expert training and advice and guidance. And of course, our PIs, Drs. Diehl-Jones and Joe Gordon, and the rest of my lab family, Matthew, Donald, Gia, and Simone. And of course, I'd like to thank CRIM for the opportunity and the funding. Thank you.
Questions for me? This might be a silly question. When you did the hypoxia, did you have a certain time length of hypoxia, then reintroduce the oxygen?
Yes.
Okay, so you had a perfusion.
Yeah, we did 18 to 20 hours of hypoxia in a very makeshift hypoxia chamber, which is a plastic bag. And then we did about an hour or two of reoxygenation. Yeah.
You mentioned neck at the beginning, so do you think your work has implications
neck?
I think that because BM3 is associated with neck, obviously there's the link there. And I believe that since we used H229 cells, they produce mucins, which makes them a more realistic, closer model to what the gut is actually like. So I think that it's a little bit more applicable that way too.
Okay, well, join me in thanking and congratulating all our speakers for today.